This is episode 55 of the Immunology podcast, Immunology 2023 on the ground. Hey everyone, this is Dr. Jason Goldsmith and Dr. Brenda Rowd. Welcome back to the Immunology podcast, where we have conversations with immunologists. The Immunology podcast is brought to you by STEMCELL Technologies, a global biotechnology company supporting life science research and fostering communication and collaboration in science. If you enjoy the Immunology podcast, rate us and leave us a review. We're always looking for feedback on how the podcast can be improved and for suggestions on guests. Today, we're bringing you a very special episode of the podcast straight from Immunology 2023, which took place from May 11th through 15th in Washington, D.C. It was a great meeting, and we had the pleasure of meeting and discussing science with researchers from all over the world. Today, we'll bring you some of their conversations as we hear from delegates about their thoughts in the meeting and research presented throughout. But before we jump into things,
Immunology 2023 may be over, but there's still more to look forward to. We are excited to announce that the Immunology podcast will be attending the International Union of Immunological Societies 2023 Congress in Cape Town, South Africa, from November 27th to December 2nd. Early bird registration is open until August 30th, and you can learn more at iuis2023.org.
all right, well, let's jump into it straight away. As we mentioned, Brenda and I attended Immunology 2023 and had a great time learning about the latest research in the field. We also had the opportunity to chat with attendees and learn about their research. So without further ado, let's introduce some of the attendees we had the privilege of speaking with throughout the meeting.
Hi, my name is Stephanie Langle, and I'm an assistant professor at Case Western Reserve University School of Medicine. In my lab, we study maternal and neonatal immunity, and specifically, we're interested in harnessing something called the gut-mammary axis, and that would be driving IgA-producing plasma cells out of the gut into the systemic and to the mammary gland that would then produce antibodies against pathogens like viruses, which is what we study, or even antibodies against commensals, which help to induce a homeostatic state in the neonatal gut. What we presented here at AAI is specifically we're looking at protection against influenza, and we're using a ferret preclinical model. And we were able to show that an Ad5 monovalent immunization was able to induce breast milk antibodies. And we are translating that to a clinical trial working with a company in South Africa to, again, induce breast milk antibodies to provide protection against viruses. So that's what the work we're doing in our lab to really potentiate maternal neonatal immunity.
Hi, my name is Seth Thomas Scanlon, and I'm a senior editor at the journal Science. I'm the immunology editor, basically, at Science. We cover pretty much everything from structural immunology to signaling of the innate immune system to mucosal immunology, et cetera. Pretty much the only thing I don't do is cancer immunology. We have another editor that handles those papers.
Hi, my name is Pawnees, and I'm a second-year master's student at the University of California, Irvine. So I am in Dr. DeQuina Nicholas's lab, and we study type 2 diabetes and polycystic ovarian syndrome, and I want to just make a disclaimer, it is recently renamed as multifollicular syndrome because there's a common misconception for people who are not in the science field and they hear polycystic, they think of cysts like cystic sacs. However, it's just referring to a bunch of follicles being made by the ovary that don't reach maturation.
Hi, my name is Abdul. I'm a graduate student from Indiana University School of Medicine. So I work in a food allergy lab, and I study T follicular regulatory cells, which are a subpopulation of Tregs. So I study the role of TFR cells in the production of immunoglobulin A, against which are reactive to gut microbiota.
Hi, my name is Shaila Mudambi, and I'm a field application scientist at Mesoscale Discovery. We work on immunoassays. So one of the, cool things about the company was during the COVID pandemic, we actually made immunoassays where you could identify whether you have immunity against COVID when you get vaccinated. So my company does a lot of that, along with, in variety of different fields, including like cancer biology, neurology, a lot of different fields. So as a field application scientist, I work with multiple different companies who use our products, and I kind of am like an experimental advisor, if you would say that. And I would go on site and I train customers, and I answer their questions and help them design experiments.
Hi, my name is Amit Singh, and I'm a staff scientist at NIA, National Institute on Aging in Baltimore. I work with the immune system. I want to understand that how the immune system basically works. And since it's an aging institute, and with aging, many things happen. The idea there is whether it goes bad, the immune system goes bad with the aging, or basically it goes dysregulated with the age. That's what we're trying to understand. In my research, basically, I focus on almost all the immune cells. It's not like understanding one type of immune cell. So I focus on almost everything.
Hi, my name is Maria Granger, and I'm a business development associate at Cayman Chemical. So my background is mostly in cytology. My undergraduate research focused on non-alcoholic fatty liver disease, and then I worked in diagnostic anatomic pathology after that for a hospital system in Michigan. Ran a lot of gynecological samples, flow cytometry. I'm really interested in learning about novel applications for flow cytometry. So I'm very interested about going back to acoustic applications for flow cytometry and developing procedural aspects of that.
Hi, my name is Tommy Lee, and I'm a senior scientist at Orna Therapeutics. Orna works on circular RNA. It's basically a circularized RNA, not a linear RNA. And we try to apply these platforms in many disease modalities. At the moment, our first concentration is immuno-oncology. We do have an infectious program as well. And now we're just trying to figure out also other disease modalities. I am on the research side, so I do a lot of the discovery. And so for me, it's more about figuring out the applications, mechanism of actions, even partnerships. These are things that I work on a lot and design a lot of the research projects that help gear towards, fruitful investments, partnerships, etc.
Hi, my name is Angie, and I'm a research lab technician at Rutgers University. I study CD8 T cells, and our lab does basically tissue-resident memory cells and non-void organs, and we just kind of stem from there on various projects. As a lab tech, I kind of have the luxury to work on different projects, but basically my project kind of goes based on previous graduate student for her role in CD103 positive and CD103 negative different subsets of T cells, and mainly specifically looking at S100A4.
Hi, my name is Amit Chimote, and I'm a research scientist at the University of Cincinnati. And our primary goal is to look at the physiology of immune cells, and we are looking at especially the roles that ion channels play on T cells and NK cells, and how they contribute to the immune cell function.
Hi, my name is Brianna, and I'm a fourth-year grad student at Case Western Reserve University. So what I study is the interaction between a protein from the coronavirus called open reading frame 3A and how it interacts with host cells. I have two projects going on right now. One of them is how ORF3A interacts with a group of proteins called the TRAFs and may lead to inflammatory signaling. The second is about how ORF3A may interact with a protein called VPS39 and how that helps ORF3A to block autophagy, a process of recycling in the cell.
Hi, my name is Damian Ramos, and I'm an undergraduate student researcher at Cal Poly Pomona. Right now, our study is with West Nile virus encephalitis, and we're looking at the roles of dendritic cells and how it plays a factor in the activation and recruitment of T cells. And so right now we're just trying to figure out what's happening with certain genes of the receptors in dendritic cells.
Hi, my name is Cindy Leifer, and I'm a professor at Cornell University. So I am really interested in how our immune system recognizes infections. And a particular subset of receptors called toll-like receptors that bind to microbial structures. But what's really cool is a subset of those recognize nucleic acids. And that's good because all microbes share those nucleic acids, but it's bad because we also share those nucleic acids. So what we're really interested in is why do we not all have autoimmune disease? How are these receptors regulated so that they're activated only when they need to be and they don't cause inflammation to our own structures like DNA and RNA?
Hi, my name is Dimitri, and I'm a graduate student at Johns Hopkins University. So I study tertiary lymphoid structures in pancreatic cancer tumors in patients that have been treated with various neoadjuvant immunotherapies. Our goal as a group is to induce more mature tertiary lymphoid structures intratumorally in these patients and do some more sort of robust anti-tumor response and hopefully see some more responders. And I study them with a variety of omics technologies, spatial proteomics, spatial transcriptomics, try to characterize TLSs.
Hi, my name is Fiona, and I'm a undergraduate at Cal Poly Pomona. Right now, I am studying the reactivation of T cells during West Nile virus infection in the central nervous system. So basically, we're looking at the perivascular space inside the brain. That's where the astrocytes are around the blood circulatory system, and in between that space is where we find dendritic cells and the antigen-presenting cells. And so what we're looking at is basically a gene knockout, and we're looking to see what it has, like the effect it has on the T cells and just the immunosuppression.
Hi, my name is Javier Rivera Guzman, and I'm a senior lecturer at the University of Maryland, Baltimore County. My primary role at the university is teaching, so I teach everything from 100 level to 600 level courses, so it's kind of broad spectrum, but my favorite course to teach is immunology.
Hi, my name is Isha, and I'm an undergraduate researcher at Cal Poly Pomona. At Cal Poly Pomona, our research lab is studying the role of dendritic cells in the reactivation of T cells in the brain in response to the West Nile virus.
Hi, my name is Janelle Montaigne, and I'm a postdoctoral fellow at the Johns Hopkins University School of Medicine. So I'm currently a postdoctoral fellow in the labs of doctors Alana Fertig and Elizabeth Jaffe. And my research really focuses on kind of incorporating multiomics in the study of pancreatic cancer clinical trial samples to really kind of promote or expedite the development of novel combination immunotherapeutics for a particularly challenging solid tumor type, pancreatic cancer. And so we're able to leverage kind of this Convergence Institute where we have computational experts and basic clinicians and immunology experts to really bring together clinical trial samples in a useful way with multiomic studies. So for example, we are looking to see kind of or find molecular mechanisms of resistance to therapies to really rationally think about the next kind of combination strategies to extend these clinical trials.
Hi, my name is Giuseppina, and I'm a PhD student at Rutgers University in New Jersey Medical School. Our lab focuses on CD8 positive tissue resident memory cells. We're more interested in the small intestinal responses. So my particular project is working on the regulation of these cells and how they will be able to respond on recall situations. I'm mostly interested in the regulation by G protein coupled receptors and different ligands that are endogenously produced, but also produced by commensal bacteria. So I'm interested in the talking situation between the gut, the intestinal bacteria, and the cells that permanently reside within this tissue.
Hi, my name is Kenny Gallivan. I'm at Case Western Reserve University, and I'm a fourth-year PhD student. So my research focuses on opioid receptor signaling in T cells and sort of the chronic impact of opioid exposure in the immune system. So what we're finding is that there's a real difference between acute exposure and chronic exposure. And so we're really trying to interrogate those differences and look sort of mechanistically how chronic exposure changes biology. We're focusing mainly on MAP kinase signaling and transcription factor modulation. Some really interesting things. So previously people have shown that there's in chronic exposure, there's an increased Th2 differentiation. And so we're trying to explore where that might come from.
Hi, my name is Laia Alsina. I'm the chief of the Pediatric Allergy and Clinical Immunology Department in Hospital Sant Joan de Déu in Barcelona, and I'm happy to be here. So my focus at work is I treat patients with primary immune regulatory disorders, which are kids that usually have like severe inflammation or autoimmunity, and these patients currently they have no good treatments to target the disease.
Well, definitely some interesting research. And we are so glad that we got the opportunity to meet all of these people. Apparently, this was the largest AI meeting yet. Did you know that, Jason? With over 4,400 delegates, which flew, or I guess also took the train into Washington, D.C. And Jason and I were so excited to be attending for the first time and representing the Immunology podcast. But I guess we wanted to also hear why delegates chose to came to the meeting and what they hope to gain. So here's some of their answers.
Hi, it's Stephanie. So, firstly, we came to present our work, and the work that we are presenting is a maternal immunization strategy to boost breast milk antibodies, and specifically trying to understand how plasma cells in the gut move to the mammary gland to produce antibodies against pathogens. And then, secondly, we came to see other individuals' talks, and what I was very excited to see is the amount of maternal neonatal immunity talks, not only from a pathogenic point of view, but also from an immune imprinting perspective.
Hi, it's Seth. I decided to attend AAI because, well, one, it's a very large meeting, and there are a lot of prominent scientists who have a lot of groundbreaking research all in one place. It also helped that this year it was in Washington, D.C., which is where science is headquartered. So it was a chance, just incidentally, to stop into headquarters and visit my colleagues.
Hi, it's Pawnees. My degree is in biotechnology, but we end up picking it's a research program. So we pick a niche and that's what we do our research in. And so I was a chemistry major in undergrad, so I had very little exposure to immunology, but that became my interest after one of our training classes. And so now that I'm in my second year, I'm here presenting a poster on my research.
Hi, it's Abdul. So this is my first AI meeting. I'm very excited about coming here just to meet the broader people of immunology and connect with them. Yeah, so this is kind of starting my immunology career from here.
Hi, it's Shyla. So right now I'm representing my company here. I'm an exhibitor. So it's kind of interesting because in the past, I've always been somebody who's attended it as a scientist or as a researcher. But being an exhibitor is very interesting because I got to hear a lot about internationally people's projects and what they're doing and what their needs are.
Hi, it's Amit. I'm a regular actually, so I always come to AI, and for me, I always enjoy coming up here. I don't look forward for something like very unique or something. I just come and see that what people have accomplished so far, and sometimes, not always, but sometimes I do feel that wow moments that I have not heard about this thing, and because of this conference only, I just came across this particular research, which is really cool, and maybe that is going to change next 5-6 years.
Hi, it's Maria. So I am an exhibitor this year, and my whole role at Cayman is to wander around, talk to researchers about what they're doing, talk to vendors about what is new and happening. So I'm very excited to be here and see what everybody's up to.
Hi, it's Tommy Lee. So what I hope to gain from AI is I appreciate the innovativeness. There's a lot of stellar research that's happening right now, very creative research that I want to kind of understand how to tailor this into the platform that we're utilizing at Orna Therapeutics. But also connect with people and also meet friends again or meet new people.
Hi, it's Angie. Here I'm supporting my fellow lab members and they're doing their presentations, and then also just being able to just go and listen to talks and kind of further gain knowledge on everything and get different aspects.
Hi, this is Amit. So, there are a couple of things that I always love about immunology. First of all, you get to hear cutting edge research, and we are primarily interested in the role that immune cells play in the tumor microenvironment. So, I attend a lot of symposiums, especially the symposiums from SITC or something about tumor immunology, and also about the cutting edge research or the new techniques. So, those are the sessions I definitely attend. And the second thing is, I like meeting the vendors because you learn new technologies, what's going on. What techniques people are doing now is so much different than what people used to do 10 years ago. So it's always an evolving field and you have to keep up with what's going on.
Hi, it's Brianna. I am here at the AAI meeting to hear about all the research that's going on, of course, but also to meet people. I'm trying to determine whether I go into industry after this or go into teaching. And I've been able to meet people who are working in both areas and hear their experiences. So along with hearing about a lot of cool research, I'm also meeting people who hold the jobs that I'm interested in for later on.
Hi, it's Damien Ramos. Honestly, I'm fairly new to the immunology just world in general. I never really kind of was involved in it before my research about 6-7 months ago. So this is all fairly new to me. And my end goal for school in general is just go to medical school. But I think getting into this research, it really blew my mind kind of like how much there is in immunology. And so like I think it was just really cool to be able to hear other people's research and other people's stories of what they're doing here, because I've never really seen the research behind a lot of things. It was more of I thought there was just things that happened and they just found things all of a sudden. But now being in this research, I know it's not easy to come across these conclusions and everything that's happening. And I think there's just so many different aspects. It's not a single person down there is doing the same thing.
Hi, it's Cindy. I love this conference. You know, I always say it's good and bad because it's a huge conference and there's so much amazing science, and you miss a lot too because it's so big. But I think one of the things that really draws me to this conference is the trainees. The trainees are giving absolutely phenomenal, clear presentations. It's just so fun to see them. And I even have a student here presenting.
Hi, it's Dimitri. Yeah, so I think it's been wonderful just seeing, people study immunology in different disease contexts. I'm a cancer immunologist, but I think it's really great seeing perspectives from people studying immune cells in different disease contexts, in different therapeutic contexts, and, sort of learning from each other.
Hi, it's Fiona. I thought it'd be a good experience, especially as an undergraduate, being able to see people like further up in their career, get a good idea, also learn more. Not a lot of undergraduates, I feel like, get this opportunity. So I think it's a great opportunity to be able to learn more. And I feel like even in general, it's just great to come to a place with a bunch of smart people. I love it.
Hi, it's Javier. I am in Baltimore, so being in DC, it was an easy way to come back to the society. It's been a while since I've been to an AI meeting, so this was just kind of an easy way to reconnect with colleagues, with people I've known in the field, to help me update myself with what's going on in the field, some of the cutting edge stuff, and to be able to just kind of network and meet new people. And yeah, it's been great.
Hi, it's Isha. So this year, it's still my first year in the undergraduate lab. So I joined very recently. And since it's a West Nile virus, neuroinflammation is a really new topic to me. And I've been learning a lot. So I came here for an experience to learn.
Hi, it's Janelle. Yeah, so I love AAI. I'm actually a 2020 recipient of the AAI Intersect Fellowship, which has been great in helping my career and continuing my computational training. And this year, I was excited. It's in Washington, D.C., so very easy for me to get to from Hopkins. And yeah, just to really kind of stay, as a postdoc, I get so invested in my own work. It's great to have come back to the basic immunology, this training that I have in the past that I kind of get lost in my own research. And it's just been fantastic to see all the amazing work that's going on.
Hi, it's Giuseppina. So actually, this is my first ever conference and my first AAI conference. So I was just trying to delve myself into the immunology field and meet people and just learn about very broad immunology and what everything is happening currently with the research.
Hi, it's Kenny Gallivan. So this is actually my first conference as a student. So it was really my first opportunity to get an opportunity to present my research to the wider community. And then as someone who doesn't quite know what they want to do when they grow up, it was a real great opportunity to network and sort of see some people's different career paths and try to start charting my own.
Hi, it's Laia. So I decided to attend a meeting. It's my first time, I have to say, and it's mostly to listen to new therapeutic approaches in the field of tolerogenic treatments.
Again, some great responses. Jason, what were you hoping to gain from the meeting?
I was trying to get an overall sense of where immunology was moving to currently. And I also wanted to just see how this meeting was run. I've been to a bunch of other types of meetings, but never been to this one. And so learning how different societies run meetings is always really interesting. I think there's a lot to gain there. We've got some more conversations with delegates in just a bit, but before we get to that, if you are going to a conference soon, or if you're looking just to expand your network, make the most of your experience by downloading our collection of tools to help you prepare for your next event. STEMCELL Technologies downloadable checklists and guides include recommendations on how to get ready before attending conferences, tips for networking, best practices for your LinkedIn profile, and more. Download the conference toolkit at www.stemcell.com/conference-toolkit.
So getting back to it, there were so many things that I was looking forward at the meeting, and obviously getting to hear about some of their great research and meeting researchers doing awesome work. But I was also really looking forward to, being in such a huge conference. I don't think I've ever been in a conference with multiple sessions and just having the opportunity of bumping into, big names here and there. Um, I obviously was very excited about all the talks in my area, cell therapy, T-cell immunology, but also the opportunity of, so looking at going for some talks that were not exactly my field, but then, for the sake of equality, trying to be exposed to a more broad range of immunological topics. So I really like the diversity of offers that was at the conference. But we were curious to see what others at the meeting were also looking for mostly. So this is what some of the attendees had to say.
Hi, it's Stephanie. So this afternoon, there is a session about immune responses over the life course. I forget the exact session title, but there are a couple talks on breast milk. So this is like SARS-CoV-2 antibodies in breast milk, but it is again looking at immune responses in the young and the old and how things change over time. So I'm very excited about that.
Hi, it's Seth. The gala tonight, because I'll be able to hopefully meet and greet a lot more immunologists, because there's only so much time in the day for meetings and going to talks, which that's the one problem that does make it a bit hard to attend the many sessions that are happening in parallel.
Hi, it's Pawnees. Obviously, I am new to the immunology field, so almost every talk that I sit in on is new for me, and I really like that. I am an auditory learner, so I love presentations and I love hearing people speak. But I do. One of the things that I really am looking forward to is sitting in on talks that are completely outside of my field. So like the cancer talks, I don't know much about it, and I think the easiest way to learn is to learn what's going on in research right now. One other thing that I think is really important is just networking. I think in this field, a lot of people are not as comfortable with speaking to one another. So this kind of gets you out of your comfort zone. You meet people that might be a field application scientist, they might be a salesperson, but everybody has this path at some point where their education background might be similar, but they might be doing different things. And a lot of PhD students and grad students who are in academia aren't aware of the jobs that are available and what they could do. And so when you're here, you can talk to the vendors, and it's one of my favorite parts.
Hi, it's Abdul. So tomorrow I have my poster session. Yeah, I'm excited about presenting my poster, talking to people, networking first to network with our immunologists and get knowledge attending different talks.
Hi, it's Shyla. So I didn't realize that there was a circulating number of posters like every day. So I think I'm really excited to see like all the new topics every day.
Hi, it's Amit. It's a pick and choose. And you have to select something and then you have to miss also something. And I think this is always true for this AI. It's not a conference, it's a fair, right? You have to, everything is going on here and there. And then, so what I look forward is I don't go to those talks which are familiar to me. I mean, so I know that work I'm doing, so I don't go to those talks. I want to go to those talks basically which have some similarity, but they have something else which they are also talking. I have generated some ideas just because of listening somebody else talk. It's some totally different, irrelevant topic, what I'm doing. And I do see that, okay, why not? If it's happening in their system, this can be also possible in my system too. It have helped that way too.
Hi, it's Maria. Yeah, I have a couple posters on my radar. I'm excited for the session that's left today. I like to wander and see what catches my eye. I look through the posters beforehand and like have an idea of some that I want, and then I always see something new that I had no idea I was ever going to be interested in, and that's what's fun for me is just stopping and talking to those people and what are you doing next? What application like struggles did you have? What worked well? What didn't? Like I don't do bench work anymore, so I miss that application a little bit, but it's fun to talk to the people who still do it.
Hi, it's Tommy Lee. Yeah, in terms of scientific, no, I think I've gotten a lot of what I wanted, but just meeting with my friends, that's really what I'm looking forward to.
Hi, it's Angie. I mean, right now I'm just here kind of taking it day by day and just looking and walking around and see kind of what catches my attention. I've been once again supporting lab members, but also from my same institution, and will further go later on today.
Hi, this is Amit. The past 2 days have been so intense. Plus, I had to give a talk, I have finished my poster and everything. Now, my brain is shut down. So, at this point, I'm done. So, I'm ready to go home. But at the same time, it was a very productive conference. I got a lot of good feedback on my poster and my talk. So, I go back with a lot of new ideas and new innovations to put, and also some papers or reviews that we can put and write. I hope to come back to Chicago next year and be a part of immunology. So, and it's a close drive for us actually from Cincinnati. So, our whole lab will be there. So, it's pretty good. And it's always a very nice. I mean, now I have started recognizing a lot of people and vendors, by their name, by their face, every time I see. So, it's pretty good.
Hi, it's Brianna. Yeah, I attended the careers roundtable that the minority committee put on Friday. And that was a wonderful experience. The table that I sat at was debating between industry and academia. So that was very fitting for my situation, trying to decide between the two. I also attended a sip and learn this morning with people interested in immunology education. I just came from a conversation with someone who does immunology education, who I met at that event. So it's been very useful.
Hi, it's Damien Ramos. I think honestly, my favorite part about it is kind of just hearing other people's poster presentations, hearing how I can kind of relate some of the things that I do with them. And I mean, a lot of the time I've spent down there looking at people's posters and trying to find out what I could do in my research to enhance that and speaking to other people so they can tell me their stories so we can kind of connect. And I think that's really cool about this community is that everyone kind of want to help each other. No one's really here to kind of show off or brag about what they've done, but they also want to learn and they want to teach as well.
Hi, it's Cindy. So I'm really looking forward to my student's talk because she is doing phenomenal work in trying to understand how TLR signaling drives type 1 interferon production and its relevance to autoimmune diseases like SLE. And so she's found this really cool role for a cytoplasmic sensor called STING in TLR signaling. So you should check it out. Her name is Carla Garcia Martinez. I love this conference. You know, I have somehow managed to go to mostly talks about innate immune recognition. Go figure. I tried to go to a whole bunch of different things, but then I kept getting drawn back to that. And there's a lot about inflammasomes. So how we regulate inflammasomes, how we activate inflammasomes, and how they crosstalk with other receptors. And that's one thing I'm particularly interested in. And so I was excited to see things like Julie Blander was talking about different kinds of LPS. So that's another structure that's bound by a TLR, and how different structures of LPS can either activate or not activate inflammasomes. So things like that, where how they're regulated, how they're signaling, the different kinases that they're activating. I'm really interested in all of that.
Hi, it's Dimitri. I'm really looking forward to the gala tonight and just, networking with a more brilliant immunologist from all over the world.
Hi, it's Fiona. Yeah, well, the conference, I'm excited to present, which I do tomorrow. And then I'm also excited to go around just Washington, D.C. in general, because it's a great area. It's super nice, super clean. And it's like there's so many things to do here. And so it's super exciting to be able to not only be at a conference, but also be in a beautiful place. So I love it.
Hi, it's Javier. Yeah, I'm checking out all the posters. I've had a few really, really good conversations. The people who are coming here seem to be on it and they know their stories and they can take good questions and give good answers. So that's always, a great dynamic to be able to have in front of a poster. Sometimes it's a story and then nobody has a question, but people here seem to be able to handle that really well. So it's great.
Hi, it's Isha. So my current mentor is presenting right now, and tomorrow my peers are presenting. So Seeing the progress that we've made in the lab is definitely the most exciting part. We're a group of five, so our lab team is a bit small. However, we work really close together. And although I have had to take a small break from the lab because of an injury, coming back to the lab has been really empowering.
Hi, it's Janelle. I'm looking forward to the gala tonight actually because A, I think it will be a fun socialization event, but B, they have apparently some collection of immunology specific kind of historic pieces to look at. And so I'm really looking forward to kind of looking at some historical immunological artifacts.
Hi, it's Giuseppina. So I'm really looking forward for Monday as I will be participating in Capitol Hill advocacy efforts for AAI, and I'm just really interested in meeting with representatives and their teams to advocate for science and a good support in the budget for NIH.
Hi, it's Kenny Gallivan. So I actually present tomorrow. So I'm really looking forward to that and getting some feedback on our project. Like I said, this is really my first opportunity outside of my institution to present. So I'm really looking forward to getting some outside perspective on, things we might not be thinking about.
Hi, it's Laia. I look forward to walk to see Washington, D.C., which is an amazing city.
Wonderful answers. So, what about you, Jason? What were you most excited about with this being your first time attending AI?
So I wanted to catch up on some of the people we'd interviewed before and see where they are at since, especially since we were able to see some of the people that we interviewed well over a year ago and see some of the progression of work. I think that was really fun. It was also, I like the exhibitor hall, but that's because I'm shopping for some instruments. And so I thought that was really useful. And for me personally, and that was really neat. And then I also really enjoyed just being in DC. Frankly, it's one of my favorite cities to have for a conference. You get to have some walking, but the conference center is nice and compact. You can go out in the evening. I know we stopped by the White House and a few other things. I think that was a blast. And I got to say the research at the meeting did not disappoint. As we know, we've covered a bunch of this, and there were so many interesting talks and posters. Let's listen to a little of what some of the attendees had to say about the coolest research they saw presented through the meeting.
Hi, it's Stephanie. So, a couple people's talks that really stick in my mind. Ailing Lim from Princeton gave an excellent talk about maternal IL 6 influencing the neonatal gut T cells, producing IL 17 and imprinting dysbiosis. Catherine Knoop, she's from the Mayo Clinic and did some really great breast milk work in clinical patients, seeing that cephalosporins reduce IgA in the mammary gland, and that could, again, this gut mammary axis link. And there were many other talks that really focused on the microbiota's interactions with mom and baby, and it was just wonderful.
Hi, it's Seth. I think a lot of people, I enjoyed the Fauci talk, his 10 lessons learned, and especially the emphasis on the need for continued vigilance. I enjoyed Sue Kake's talk on metabolic control of CD8 T cell exhaustion. But also in that same session yesterday, there was somebody named Timothy O'Sullivan that was looking at what drives sex differences in NK cell effector function, which I thought was quite interesting. I think that's a really interesting topic, sort of sex differences in the immune response. And Ing Lim gave a really nice talk yesterday about maternal IL-6 signaling in utero that imprints fetal epithelial stem cells and leads to lifelong changes.
Hi, it's Abdul. So, my favorite talk was the Dr. Forge's talk the other day.
Hi, it's Shyla. Yeah, I think with immunology, a lot of times we focus on cancer therapies, like T-cell therapy is something I'm very familiar with. But what I really liked was the focus on autoimmunity. I have kind of a personal connection with that because I've had a lot of friends who've randomly figured out that they're allergic to certain things or they're feeling fatigue from certain types of foods. It's really nice that there's also another focus on autoimmunity.
Hi, it's Amit. I think whatever I'm going to say, maybe everybody have said that. Definitely yesterday when we listened to Fauci, it was, I have heard him many times. It's not the first time I was listening him, but every time I listen him, I feel like there is, you see the energy which he have and the concept which he presented yesterday, the 10 lessons which we learned from pandemic, it's an amazing thing. And many of those we already know, but when you listen it from Fauci's voice, it have totally different meaning. And to me, it's a really, really powerful message. we need to be ready for next pandemic. I think that was one thing. The other talk, which actually I was looking forward for that, is Shane Crotty. So I follow his research too, and yeah, this is really cool. And there have been some, some speakers like students also. They have really done very good presentations. So yeah, overall, once in a while I do listen something which will keep me awake for a couple of nights. Yeah. After pandemic, and I think Fauci was also pointing out, and many people actually do feel that way. I think science communication, which you guys are already doing that, I think it's a need nowadays, and responsibility also. I think if you have any affection to science, I think it's good to have science communication in a proper way, and in a language which people can understand.
Hi, it's Maria. I visit a lot of people that I work with on the regular basis. One application that I was really interested in is I met some people working with CB2 antibody receptors, which Chemical does some forensic applications, and that's usually what I hear about is mass spec. And it was interesting to see like a cellular view on cannabis and THC. So I'm interested to see what happens there.
Hi, it's Tommy Lee. So for me, as you already know, I have focuses that I have to go to, but my internal passion is autoimmunity. So I've been attending a lot of these autoimmunity talks, especially the sessions that were guided by Brian Evervold from University of Utah. And a lot of those talks that were there, a lot of those academic talks were just very creative in what they were discussing. And so I think that's the session I remember the most from my AI moments.
Hi, it's Angie. I mean, it's nice to always get more insight on CD8 tissue resident memory cells, but also just, I think, looking at other various parts of the body that these tissues kind of are found and reside, and not just where we study in the gut mucosa or like spleen, but just in like lung, or looking at different techniques that some students are looking in. And I think today my favorite one was looking at an ex vivo lung transfusion and just looking at tissue resident memory cells there. I think that was my favorite one.
Hi, this is Amit. I love all the sessions on the tumor microenvironment, but I especially liked the session that was by SITC yesterday morning, that the Society for Immunotherapy for Cancer. And in that, researchers were highlighting in developing novel targets and how those can be used for immunotherapy. Because as you know, immunotherapy is a field which is quite evolving, but sadly, in most of the tumors, it's effective only in 15 to 20% of the patients. So this kind of research is extremely important. And I want to also say thank you for using this platform for communicating science, because many times researchers do their research, but what's the use of what we do in the lab if we can't communicate it to the general population?
Hi, it's Brianna. So I am from a structural biology lab, and I attended Hao Wu's talk last night about all the structures that she has solved. And I knew that she was a big person in the field before this, but just like seeing on one slide all of the structures that her lab has worked on was really impressive, and just hit home like, wow, this is a person who has done a lot for the field. This is my first conference away from my home institution, and it's just been a very, very good experience. A lot of positive interactions with people and good research. So I'm hoping to come back for sure.
Hi, it's Damien Ramos. I'm really into neuroscience. So I liked a lot of the talks about infections within the central nervous system and how it progresses, and like what actually happens inside of the brain, because there's just so many different elements and how it can affect so many different body systems.
Hi, it's Cindy. Well, they brought Tony Fauci. How can you beat that? I'm probably everybody's saying the same thing, but I would say that was just a knock your socks off, amazing, clear talk with it was so powerful. So, how can you beat that? But no, I think, joking aside, they always have sort of the same structure where you've got these main, symposia. But what I think was really cool, I was talking to a couple people. They tried to really get not the huge big players, but get those up and coming scientists to talk at those symposia. They'd bring one of the big keynote, big speakers that drives a lot of people into the room, but then gave the platform to a lot of up and coming trainees or a lot of up and coming assistant and associate professors. So I thought that was really good.
Hi, it's Dimitri. Definitely Dr. Fauci's was was incredible, but there were a couple talks that I really enjoyed of people looking at viral specific T cells in tumors, in brain tumors, for example. I think that's something that we're also interested in looking at, in terms of how tertiary lymphoid structures are developed. We're really interested in seeing what's the connection between viral responses and sort of like tumor progression.
Hi, it's Fiona. I have to say, Fauci. I feel like you have to, he's here. We're in Washington, D.C. Not everybody gets to, see almost like the man, So he's a man of immunology. So I have to say, probably he would be the best one.
Hi, it's Javier. This morning's session on myeloid cells was super, super interesting. I thought some of the work that was presented was great. There was a talk on the NLRP3 inflammasome from a biochemist who was doing some physical biochemical work on that, and that was really impressive. That was amazing. That was really cool stuff. So I checked out some of these. The one that comes to mind, especially, is the one with gaming, and so I got some really nice ideas of what to do. There was a researcher or instructor who had created a board game for immunology, right? And so gave me ideas on what I might be able to create in terms of just kind of adapt that and for my purposes. And I think that'd be great. If you can make it a game, you're learning and having fun at the same time, and you get the best of both worlds. Yeah, at this wonderful conference, I hope more people start coming so we can train the next generation of immunologists. And yeah, next year I'm hoping that I can submit an abstract so I can do something on art and immunology, because that's something that I do in my classes, so that we can kind of maybe expand that realm a little bit.
Hi, it's Isha. So learning more about viruses and different immune response has been really eye-opening, especially with the T cell activation. Well, I hope to be presenting at AAI next year. I have a year left of college, and I hope to make the most of it.
Hi, it's Janelle. I think probably everyone says Dr. Fauci, first and foremost. That was an incredible talk. But more specifically, I've been really interested in the immunotherapy type research talks. And there was one in particular, I forget the presenter's name, but they were looking at how CD38 kind of promotes terminal T cell exhaustion, and actually knockdown of CD38 in a mouse model kind of abrogated that exhaustion phenotype, which I think is fascinating. Kind of these alternate mechanisms of T cell exhaustion are really crucial, especially in the context of human clinical trials too, getting back to that biology of what can you treat to help patients. There was another great talk that I saw about sex bias and kind of T cell exhaustion in cancer. And I'm not sure if you covered this already, but they looked at kind of AR and how it promotes the transcription of tox, I think it was, to promote T cell exhaustion, which may kind of help explain some of these phenotypes of, men having more severe cancer outcomes than women. And yeah, I'm trying to think. I was just at a great talk going down the infectious disease route, where they were actually looking at these heterotypic nanoparticles that you can use to vaccinate people against, SARS-CoV-2 and related viruses using very conserved receptor binding domain regions.
Hi, it's Kenny Gollivan. Yeah, well, Dr. Fauci's talk last night was fantastic. But in terms of like science, the actual special lecture last night from I think it was Douglas Green, where they were looking at sort of asymmetric cell division and how that influences T cell fate as early as that first division. It was really, really interesting. The fact that they can track daughter cells in that way identical daughter cells is a really cool application of the technology. So it's a lot of fun. I haven't been here for a while. I think it was like an 8th grade class trip. And so it's cool. It's a really cool city.
Hi, it's Laia. So there was a talk that I really was impressed with, and that was on Friday. It was related to using the liver as a tolerogenic platform to generate tolerogenic responses to particular antigens, and they are using this platform for celiac disease. And I was thinking that probably if we use that for other diseases that have an antigen driving the pathology, then I think we can do that. And it was on Friday, and it was a very nice talk. So my experience here has been great because I've had a lot of time to discuss with other people in the field. So after the talks, everybody was staying nearby, so you could find a moment to discuss the talk with the PI. So I thought it was really very nice for networking.
So I have to agree with a number of those responses. Really nice talks. I guess that personally, my favorite session is, it's hard to tell what my favorite session was, but I really enjoyed the first session we attended on that Thursday, day one, maybe because I was very fresh out of, starting first day of conference. It was on B cell and T cell helper responses during viral infections. And I thought it was really nice just to see the cooperation between them. And then I also like on the last day, on the Monday morning session at 8 a.m., there were not that many people, but it was about immunity to emerging pathogens. And we saw, going back to your point of like seeing where our guests are now, we saw James Crowley from Vanderbilt University presenting about the development of monoclonal antibodies against SARS-CoV-2 and other emerging pathogens. And it was so interesting because we interviewed him like almost 2 years ago. And it was also followed by a discussion from AstraZeneca from 2 of these monoclonal antibodies that his lab identified. They made them into therapy. So it was a whole journey of the antibody. It was very interesting. So I think I really liked it. I was very tired by then, and there were very few attendants, but I'm glad I went.
Well, that brings us to the end of our series of Immunology 2023 episodes. We had a great time at the meeting and hope that you enjoyed hearing from some of the attendees. Don't forget to check out the episodes that we released during the meeting and subscribe to our newsletter at www.immunologypodcast.com to get the show notes, including an episode summary and links to all of the interview and roundup papers. You can also reach out to us on Twitter at @immunopodcast or via email info@immunologypodcast.com with feedback or to suggest guests. See you next time.